It is now my great pleasure to introduce Dr. Mary Brunkow, our second laureate for this afternoon. Mary Brunkow was born in Portland, Oregon, where she earned a Bachelor of Science degree in Cell and Molecular Biology from the University of Washington. She then transitioned to Princeton University, where she completed a PhD degree in Molecular Biology in 1991. She then moved to the Samuel Lunenfield Research Institute in Toronto for postdoctoral training. In 1994, Dr. Brunkow joined Darwin Molecular Corporation, a gene discovery biotechnology startup company in Bothell, Washington State. It was co-founded by Leroy Hood. Brunkow remained at Darwin after it was acquired first by Cyroscience and then by Celltech. And she remained there until the site closed in 2003. A few years later, Dr. Brunkow joined the Institute for Systems Biology in Seattle, and where she is still working as a program manager in genetics, and she coordinates multidisciplinary projects that integrate human genetics with whole genome sequencing and computational approaches with the overall aim to understand complex diseases and make discoveries of medical importance. She's currently actively working to promote education and access to education and training for young women through mentorships. And hopefully this platform that she now has will help her in these pursuits as well. So please join me to welcome Dr. Mary Brunkow to the stage to give her Nobel Lecture 2025.
Thanks very much for that introduction, and thank you all for being here. I especially want to thank the Nobel Assembly and Nobel Committee of the Karolinska Institute for this great honor. In the past couple months since the announcement of the Nobel Prize, I've had a lot of time to think back on the work that we did 25 to 30 years ago. And I realized that I wouldn't be standing here today if it weren't for a couple of really important mentors in my life who showed me what a life in research would look like. And the first one of these is Dr. Larry Sandler, who was a fruit fly geneticist in the genetics department at University of Washington, where I was an undergraduate. And I was already pretty excited about genetics when I was in school there, but when I attended an upper-level genetics course with Dr. Sandler, I was especially enthralled by the way he wove Drosophila genetics lore into whatever topic he was covering in his lectures. And then I was really fortunate to be able to do an undergraduate research project in his lab. I spent about 1.5 years in his lab, where I saw what being in a research environment was like, and research was not really anything that I had on my radar as being a potential career track. I thought I wanted to go to medical school, but after spending a lot of time discussing with Dr. Sandler what my future might bring, I ended up going off to graduate school at Princeton University, where I was very fortunate to do my PhD research with Dr. Shirley Tillman, who is an extraordinary scientist and a fabulous role model, both for young women in science, but really anybody who's in her orbit. And working in Shirley's lab really taught me how to think critically, but also keep an open mind in science and in life. So I'm also really lucky today to have been preceded by the world's expert in immune tolerance, so I'm not going to spend very much time introducing the topic, just give a quick overview to remind you that immune tolerance is the process by which the immune system keeps a balance between T cells that recognize non-self antigens that would be harmful to the organism, These antigens could be presented by pathogens or environmental pollutants, and they are recognized and neutralized by the system. But the immune system also has to be able to tolerate self-antigens and not destroy the resident tissues in the organism. When this system becomes unbalanced, you see things like an autoimmune disease and the resulting tissue destruction, et cetera, as the T cells attack self-tissues. When we entered this area of research, it wasn't necessarily to contribute to understanding of immune tolerance, but we were more generally interested in identifying targets that would help us develop drugs to treat things like autoimmune disease or cancer or transplant rejection. And the vision that we were starting with was using genetics as a way of getting new insight into this particular biological area of research. And the story starts with this mutant mouse that we were interested in. And it begins in the Oak Ridge National Laboratory, which was established in Tennessee in 1943 as part of the Manhattan Project during World War II. So while most of the efforts in the laboratory were focused on developing the atomic bomb, there was also quite a bit of interest in understanding how radiation affects people and other mammals. And so a mammalian genetics section was established, and William and Leanne Russell, who were really top-notch mouse geneticists from the Jackson Laboratory, were recruited to Oak Ridge to head up this unit, where they were tasked with studying the hereditary effects of radiation and radiation-induced mutation rates. So the Russells did some really important experiments that did, in fact, contribute to our knowledge about the effects of radiation. But kind of a bonus for them as mouse geneticists is that they ended up with this huge collection of really interesting mutant mice, which over the years has been a tremendous resource for the scientific community in identifying new genes that are important in a number of different biological areas. So in 1949, they observed this mouse mutant that they called Scurfy, mostly just because of the way it appeared. And interestingly, this is a spontaneous mutation that actually didn't arise in any cohort of mice that were exposed to radiation. And they maintained this strain for about 40 years before anyone really was able to study it in any depth. They just knew that it was something important because it has this really striking lymphoproliferative disorder. It's also the first X-linked mutation reported in mice. So just when you look at these mice, so on the left is a Scurfy mouse, so a male mouse with a single X chromosome that's carrying the Scurfy mutation. They show this kind of general wasting phenotype and a scaly skin that looks a lot like psoriasis or eczema. When you look internally, there's huge enlargement of the spleen, liver, lymph nodes, and this is due to infiltration by lymphocytes. The little Scurfy mouse is sitting next to his sister, who is a carrier for the Scurfy mutation, but there's really no phenotype in females. So in the 1980s, Virginia Godfrey and Irby Wilkinson came to Oak Ridge, and they performed kind of the first studies of the phenotype in Scurfy mice. And they were able to show that this phenotype is mediated by CD4 positive T cells. They showed a marked cytokine elevation and concluded that this autoimmune syndrome is a result of a defect in the downregulation of T cell activation. So it seems to be really an incredible model for autoimmune disease. Now, fast forward to the 1990s, And what was happening at that time was kind of an explosion in new approaches and technologies and computational methods that were going to allow us to characterize the whole genome. So looking at all genes, gene structure, chromosome structure. The Human Genome Project was launched in 1990. And in 1993, this small biotech startup company, Darwin Molecular, was launched just outside Seattle, Washington. And the vision behind Darwin was to use genes as a way of discovering novel drug targets. And the initial areas of interest were in autoimmune disease, cancer, and AIDS. And interestingly, so I'm showing, let's see, these are the founders of the company. And David Gallus, immediately prior to coming to Darwin, moving to Seattle, he had been the director of biological and environmental science at the Department of Energy, which was the major funder for Oak Ridge National Lab. So he was very familiar with the mammalian genetics unit and well acquainted with the Russells, the geneticists, and he had a real appreciation for the power of mouse genetics in helping us really dive into particular biological areas. So David facilitated collaboration with the Oak Ridge group, and we decided to work first on figuring out what the molecular basis of the Scurfin mutation is. And being a startup company, there was definitely that feeling of, everybody wears multiple hats. Here's a picture of David pushing a broom through a lab. He was also the president and chief science officer of the company. So I joined the company in 1994, and my background was in genetics, genomics, molecular biology, and I was tasked with finding the mutation that causes the Scurfin phenotype. And we decided to take a two-pronged approach to cloning Scurfin that would involve first genetic mapping. So we knew the Scurfin locus was on the X chromosome, and some sort of general localization had been performed already, so we had a good starting point. But I decided that we would just continue to let the mouse do the work for us and start trying to narrow that interval further by scoring for recombination events that would help to narrow the Scurfin locus. So we brought the Scurfin mice to Darwin in Seattle, but first we had to find a home for them, and so that was an underutilized janitor's closet with a sink and a self-contained mouse caging system. And I was able to set up a big backcross experiment in that janitor's closet in order to score recombination events. And this, oops, sorry. Oh. I don't know if I'm going backwards. This is just a copy of a piece of paper that I had up on my bulletin board for years, it seemed like, where I was tracking recombination events and also trying to compare our X chromosome mapping to other maps that were being published at the time. It was a very confusing time where there wasn't, you have to understand that there was not really genomic sequence available at the time, and so but everybody was really trying to put together maps showing orders of markers, etc. So we combined the genetic mapping with physical mapping. So we wanted to ultimately really understand at the DNA level, at the nucleotide level, what was happening in the region that contained the Scurfin locus. And as I said, as sort of an outcome of the Human Genome Project, there were a number of really, cutting-edge technologies that were available, and some of these are listed here. And I think it's worth pointing out that our situation was perhaps unique. Being in a biotech company, we had the funding that allowed us to incorporate a lot of these different resources, as well as the mouse genetic breeding experiment as well, which is all very expensive. It would be difficult for an academic lab to do all of this simultaneously. This was my core team that worked closely with me. Eric and Catherine were really very talented molecular biologists, worked great in the lab. Brian Paper was a very talented DNA analyst and bioinformaticist who had a lot of experience with both staying on top of what DNA sequences were available in the databases, which was pretty limited at that time, but also being really aware of new analytical tools that were coming online. So the process that we went through for finding the Scurfin mutation involved, as I said, the genetic mapping. We ultimately found, so the red X's on this chromosomal region represent the most informative recombination breakpoints that we identified. And at the same time that we were doing the genetic mapping, we were using whatever molecular markers we had in that region to start pulling out large cloned fragments of genomic DNA and kind of generating a tiling path or a contig of artificial chromosomes across the region. And in the end, we were able to estimate that the physical region was about 500,000 base pairs long. And once we had a number of cloned sequences across the region, then we started doing really high quality DNA sequencing of them. So we had a really good core facility to do the DNA sequencing ourselves in-house. Then once we had that really high quality genome sequence across the region, we were able to apply whatever kind of analytical tools and algorithms that were available at the time. And we ended up with a list of about 20 genes in the region. So just by blast analysis, you could find homology to known genes for certain. So about half of our genes in the region were previously reported genes. But then tools like GenScan were actually quite good at predicting novel coding sequences. So they would spit out, exon intron structure and find 5' transcription start sites and the like. And this is just what the output from GenScan looked like when you fed it, a couple hundred KB of genomic sequence. So we had a list of 20 genes, like I said, and then the trick was just to sequence, and this was all sequence, of course, that was derived from the wild type version of genomic DNA. And so for each of those genes, we cloned and sequenced the Scurfin version of the gene, looking for a mutation. And where we found the mutation turns out to be this gene here that we were calling FKH. It was a novel gene that had never been reported, but it did have homology to a family of fork head domain containing DNA binding proteins. And this is a page out of Catherine's lab notebook that shows that reports the mutation. I'm not going backwards very well. But anyway, so this slide here is kind of our overall summary then of the region, which shows the gene content. Again, these X's represent the most informative recombination breakpoints. And then I wanted to show this nice sequencing chromatogram, because we never look at those anymore. But this was a really nice example of that clearly shows an insertion of 2 extra adenines in the Scurfin versus wild type. And this mutation, of course, would cause a frame shift in the translation of the mRNA. And it's located in the DNA binding domain of this novel fork head protein. So as soon as we sequenced as soon as we figured that we had the Scurfin mutation, that big celebration all around, but then we realized that there was going to be quite a bit of validation and confirmation of this. But we also, at the same time, right away cloned and sequenced the human ortholog of this sequence, this novel sequence. And then we submitted those sequences to a nomenclature committee because the protein was part of this known forkhead domain family. This is what was expected. At the time, you would submit sequences and the committee would do a phylogenetic analysis to determine where within the family this novel sequence lies. So then the FOXP3 name was assigned to our sequence based on where it fits into the whole tree of forkhead domain containing proteins. One of the ways that we confirmed that we had indeed cloned the Scurfy gene and that FOXP3 was Scurfy was to use a transgenic mouse approach to see if we could rescue the Scurfy phenotype if we provided the wild type version of the gene. So we subcloned a large genomic fragment that had only our novel coding sequence on it, along with some 5' and 3' sequence, with the hope that this transgene then would mimic or recreate the same expression pattern that the endogenous FOXP3 gene has. And so this purified DNA was microinjected into mouse oocytes, and we generated 5 independent lines of transgenic mice carrying the wild type version of the FOXP3 gene. And in those 5 lines, there was quite a wide variety of copy number, which was interesting because those overexpressing transgenic mice also have a phenotype, which Dr. Ramsdell will get into in more detail in a moment. But what we found, well, first of all, what we found was that male mice that carried the Scurfy mutation but also had the transgene, so we bred the transgene onto the Scurfy background. And those male mice were completely healthy. They had, normal lifespan. You couldn't tell they had any phenotype at all, which was also strengthened by what we saw internally looking at organs. And so this just shows what a Scurfy lymph node normally looks like with the great infiltration of lymphocytes compared to wild type. And you can see that when you combine the transgene and the Scurfy mutation, they look basically like wild type. So we were really convinced by this that we had, in fact, cloned the Scurfy gene. So we wrote that up and reported those results. But then we also were aware that there was a human condition that also mapped to a similar region on the X chromosome and shared a lot of features in its presentation with what a Scurfy mouse looked like. And we collaborated with a couple different clinical genetics groups. So Hans Ochs group at the University of Washington and also Bob Wilden in the Oregon Health Sciences University. They had been studying families that have this IPEX syndrome. And we were able to get DNA from them, from the patients and families that they had been studying, and we indeed found that the human ortholog of FOXP3 was mutated in this condition. So we could demonstrate that FOXP3 is the underlying basis of IPEX, and IPEX is an acronym for all these different syndromes. And so basically, again, a very severe autoimmune syndrome in young boys. It's usually seen very soon after birth. And interestingly, in contrast to what we have from the mouse, where we had just a single mutation in the Scurfy, with IPEX, now there have been dozens of mutations reported, which kind of gives you a nice allelic series in the gene, and mutations of all different types that affect different portions of the protein. And it has been reported that there's a pretty good correlation between the type of mutation in the gene and the severity of the disease. So there's a lot of variability in the way that IPEX presents itself. And this is significant Because, well, first of all, these days, as soon as a child is suspected to have IPEX, the FOXP3 gene is sequenced, which gives the clinician a lot of information about whether, I mean, it's an important diagnostic feature. And then, like I just said, since there is some correlation between the type of mutation and the severity of the disease, that also gives the clinician some prognostic information. And from a molecular biology point of view, all these different mutations really have helped people analyze in great detail what the importance of different functional domains of the protein are based on what these mutations are doing. So, as I mentioned, FOXP3 is a DNA-binding protein, and it is a transcription factor that controls a lot of downstream genes. It's a novel member of this fork-head winged helix family of proteins, highly conserved across all vertebrates, and it can act as either a repressor or activator depending on the other proteins that it interacts with. This is a very simplistic cartoon just to highlight some of the main functional domains in the protein. So I wanted to talk a minute about kind of what this means from a drug development point of view. So remember, we were working in a biotech company where the ultimate goal is to develop a drug that can be used in the clinic and help patients. And I kind of see two different scenarios coming out of an approach that, like what we were taking, where we started with an interesting phenotype. We want to discover a gene, but we have no preconceived notion about what the function of that, or what that gene or protein is going to be when we first start this kind of a study. So there's kind of two different scenarios that can come out of that. And the first is where the gene that is discovered doesn't really have direct human health implications. And what I mean by that is that the protein is maybe not particularly druggable itself. And FOXP3 is a good example of that. Being a DNA binding protein, it's in the nucleus. It's pretty hard to access with, a small molecule or a biologic type drug. But I'll get back to you. I mean, it doesn't make FOXP3 any less interesting, but it is hard from a drug development point of view to figure out how you could directly manipulate FOXP3. The second scenario is one in which the gene discovery that you make actually does lead to a protein that is accessible and a good drug target. And I just want to take a short detour and describe a project that I was also involved in that was happening in parallel with the Scurfin project. And this is one where we discovered a gene called SOST. And in this case, what we started with was a human condition called sclerosteosis, which has a very strong effect on bone density and bone growth. It's an autosomal recessive, so presumably a loss of function of some important gene. And what's shown here are X-rays of skull and hands from a normal adult compared to what you see in an individual affected by sclerosteosis. And you see this, really tremendously increased bone density. It's characterized by excess deposition of bone throughout life. It's very strong, as in contrast to some other conditions that affect bone density. The quality of the bone is actually very high, and all different bone types are affected. And there's very little other pathology that's not bone-related in this condition. And carriers, interestingly, so if you're heterozygous for sclerosteosis, there's really no apparent visible phenotype, but they do have an intermediate bone density, which implies kind of a dose-dependent, which is very nice from a drug development point of view also. So we went through a similar positional cloning exercise as what I just described for Scurfin, but in human families affected by sclerosteosis. And we, at Darwin, at the time, we were kind of working under this idea of a surrogate drug approach. And what I mean by that is that we were specifically looking for phenotypes in mouse or human which under certain conditions would be a clinically relevant phenotype that you might want to actually use to treat a disease. So what I mean by that in this case is that in sclerostosis, this loss of function mutation is causing a big increase in bone density, which would be exactly what you would want to do in an adult that's affected by low bone density. So like in the case of osteoporosis or chemotherapy-related bone loss, if you could kickstart bone development in an adult, perhaps you could, really increase their quality of life. And so at the end of our positional cloning effort, we identified a gene called SOST. And this was very exciting on a number of levels. First of all, it was a novel gene, which was great for the company from an intellectual property point of view. It's secreted, which does in fact make it really accessible to small molecule or biologic type drug that could actually interact with this gene product and modulate its effect. And it's working in a biological system that was already fairly well understood. So it's an antagonist of BMPs, or bone morphogenic proteins. So it was an area that where there was already a lot of knowledge. So this was an example of a really successful outcome from this kind of gene-based drug target discovery approach that the company was doing. But back to FOXP3. Oh, and the most important point, of course, is that there is, in fact, a blocking antibody now that's on the market. There is actually a drug that's being used to treat osteoporosis, and this was co-developed by Celtec and Amgen. So back to FOXP3, and I'm pretty much wrapping up here. I want to just summarize some of the things that we know about FOXP3, and to get back to the point of that FOXP3 itself as a protein is a hard one to approach from a drug development point of view, but it being a transcription factor, in fact, opens a whole world of downstream targets and other genes that regulate FOXP3 itself. And in fact, once this work was published, there's many genes or many labs around the world who have in fact made great progress in understanding the whole kind of gene regulatory network that FOXP3 works in. FOXP3, and this is kind of an extension to what Dr. Sakaguchi was talking about, we were able to show that, so we were aware of these suppressor or regulatory T cells that Dr. Sakaguchi had reported on, the CD4 positive, CD25 positive cells, and we were able to show that FOXP3 is really very specifically expressed in that very small subset of CD4 T cells that are also expressing CD25. And these really define the regulatory T cells that we've heard about today. So FOXP3 was a really important marker and kind of gave researchers a hook into this whole new field of regulatory T cells. We now know also that FOXP3 actually programs or directs the differentiation of CD4 positive T cells into the Treg pathway. And there are now a lot of interesting advances that are related to harnessing the power of the Treg cells, sort of separate from the FOXP3 protein itself, but the Treg cells that are defined by FOXP3. We've made a lot of progress in autoimmune disease, organ transplantation, and cancer. And again, you'll hear more about this from Dr. Ramsdell, which is exciting. Definitely a big team effort. So the folks at Darwin Celtec are listed over on the left with kind of our key team members in bold, but really there were so many people within the company who contributed at various levels to this study. Our collaborators at the Oak Ridge National Labs, and then the clinical geneticists at University of Washington and Oregon Health Sciences, we owe a lot to. And then, you know, I obviously am not working with the people at Celtech Darwin anymore because the company doesn't exist anymore. The doors closed in 2004, but I have been working at Institute for Systems Biology for a number of years, and I must say that, you know, since the announcement of the Nobel Prize, I've gotten just, you know, loads of support and encouragement from people at ISB, and I just want to call out especially Jim Heath and Lee Hood, who have really helped a lot, you know, sort of navigate this new life that's happened since two months ago. And just as a quick, you know, indicator of how close-knit the team was at Darwin Celtech, a couple weeks after the Nobel Prize announcement, you know, this big email went out to every former employee that we could contact, and we had a reunion, pretty, you know, short notice at a bar in Seattle, and you can see there was great enthusiasm and quite a good turnout of people showing up to celebrate this great news. So with that, thank you.